Hello everyone. Today I'm gonna talk to you about one rather complicated topic, and because of its complexity and because of a huge number of information that I would like to share with you this time, I'm gonna divide this lecture into two videos. Today we're gonna talk about acid-base equilibrium, first part, and the second part. In this first video we're gonna start and we're gonna do some of the mechanisms of acid-base equilibrium, and then we're gonna continue with the upcoming video to complete the story on acid-base equilibrium. So what we all know is that actually water makes up most of the total body mass, and that is the information with which we have started our biochemistry course at the very beginning of the semester. But still what we know is that water, of course, is not found in its pure form, but rather in the form of a solution of various molecules and ions. So body fluids have to meet the following requirements so that the life processes can proceed normally, and those requirements are constant volume, constant osmotic pressure, constant composition and content of dissolved ions, and what is going to be our target for today, constant concentration of hydrogen ions, or constant pH of body fluids. This is the very important function of the organism, the maintenance of the constant concentration of hydrogen ions. Because the slightest variation in these concentrations or the slightest variation in pH of body fluids may cause the significant change of the rate of metabolic reactions in cells catalyzed by different enzymes. So you all probably know that the approximate pH of body fluids and blood is 7.4, or the concentration of hydrogen ions is approximately 40 nanomoles per liter, with a physiological range between 7.36 and 7.44. Everything outside this range is considered to be a pathological value. The range between 7.3 to 7.52 can be connected with life, and it corresponds to approximately 50 to 30 millimoles per liter. Everything else has a lethal effect towards cells and organism, of course. But what about the origins of acids in the body? So we know that carbohydrates, lipids, and proteins can be degraded or can metabolize in a number of oxidative reactions, yielding the formation of different acids. So certain systems and mechanisms are required to neutralize those acids and to maintain the constant pH of extracellular fluids. So let's make an overview of the metabolism, and let's see which are those acids produced during metabolism. So during carbohydrate metabolism, this is what we know. Glucose is degraded to pyruvate in aerobic glycolysis, which is the salt of pyruvic acid. In anaerobic conditions, it's metabolized or reduced to lactate, which is the salt of lactic acid. On the other side, pyruvate undergoes oxidative carboxylation to acetyl coenzyme A, which enters Krebs cycle, finally catabolizing glucose to carbon dioxide and water. And you know from your chemistry course that carbon dioxide and water tend to form carbonic acid, which is of course an acid. What about TAG metabolism? 3-acylglycerols are hydrolyzed to glycerol and fatty acids, which are weak organic acids. Fatty acids can be further metabolized to give carbon dioxide as well, or carbonic acid as the final product. Ketone bodies are also formed, like acetoacetic acid or beta-hydroxybutyric acid, and we said that those are medium strength organic acid. What about the metabolism of proteins? Proteins are hydrolyzed to amino acids, so they are acids. They can catabolize further to give some of the intermediates of Krebs cycle, thus they can be catabolized to carbon dioxide and water, which means carbonic acid. Some other amino acids containing sulfur, for example, in a side chain like methionine and cysteine, they can undergo metabolism to produce salts of sulfuric acid, so again an acid. Nucleoproteins can metabolize in order to produce acidum uricum or uric acid, and organic phosphates are catabolized to hydrogen ions and phosphates. So whichever way you choose of metabolism, at the end, as the final product, you get the formation of certain acids and their salts. So those numerous types of acids are classified in two groups, non-volatile and volatile. Non-volatile acids are, for example, phosphoric acid, sulfuric acid, lactic acid, pyruvic acid, acetoacetic acid, beta-hydroxybutyric acid, and numerous others. Volatile acid is physiologically important carbonic acid, and it's called volatile because it can be eliminated in the form of carbon dioxide as its gaseous form. So the complete reaction and equilibrium of carbonic acid is represented on the slide. So carbonic acid is rather unstable, so it tends to be in an equilibrium with the carbon dioxide and water. But on the other side, it's considered to be a weak acid, so it can dissociate to bicarbonate ion and hydrogen ion. In vitro systems, the reaction and disequilibrium and those reactions are spontaneous, but in the body we have just a few spontaneous transformations. Most of them, like this one as well, is catalyzed and regulated by enzymatic activity, and this time this equilibrium is regulated by carboanhydrase activity. There are several isoenzymes of carboanhydrase, and we're going to mention those isoenzymes later on during this topic. So, in order to translate these theoretical overviews into numbers, so we can say that acids are formed continuously in the body. So during one day, in 24 hours, approximately 20,000 millimoles per liter of hydrogen ions are formed and synthesized from volatile carbonic acid, and approximately 80 millimoles per liter of hydrogen ions is obtained from non-volatile acid. So it's a huge amount of hydrogen ions which has to be neutralized by certain mechanism. Those mechanisms obviously list the chemical buffers or physiological buffers as chemical systems which are used to prevent the change in pH value, but also some organs like lungs and kidneys are also involved in such a regulation. So by the term acid-base equilibrium, we mean that this is the topic which involves maintaining a constant pH of body fluids. And this is achieved by very efficient mechanisms, because if you take into account an enormous quantity of hydrogen ions which are synthesized, you have to have a perfect mechanism for neutralizing such a huge amount of hydrogen ions normally produced during metabolism. So the following is essential for maintenance of constant pH of body fluids. So first, chemical buffers, definitely first defense line. So they serve to neutralize excess amount of hydrogen ions. Then we have lungs, which role is to eliminate carbon dioxide, and kidneys, which role is to excrete hydrogen ions and at the same time control levels of bicarbonate ions by maintaining the equilibrium with carbon dioxide and water. So we're gonna start step by step. First, we're gonna introduce chemical buffers and their properties in maintaining of pH, and then we're gonna talk about respiratory control, and then we're gonna continue with renal control. So this first part of the lecture is gonna focus on those two mechanisms, first two mechanisms, and then in the part two we're gonna focus on renal control of pH. So physiological or chemical buffers are maybe the most important mechanisms. So we have a few very important physiological buffers, and I hope that they are very well known to you, so we're gonna make an overview. So hemoglobin and hemoglobinate with a pK 5.4 to 9.4. Then we have protein, proteinate with the same range in pK values. Carbonic acid and bicarbonate, which form carbonic buffer, bicarbonate buffer with a pK 6.1, and phosphate buffer, which contains dihydrogen phosphate and as acidic component, and hydrogen phosphate as a conjugated base with the pK approximately 6.7. But those buffering systems are not equally distributed in all body fluids. So in plasma, in blood, we have an activity of bicarbonate buffer, and we have blood plasma proteins with the same role. In interstitial fluids, we have only bicarbonate buffer active because this compartment, extracellular compartment, lacks proteins. In erythrocytes, we have all of these buffer active. So hemoglobin buffer, protein buffers, bicarbonate buffer, and to a lesser extent, phosphate buffer. Phosphate buffer is not that active in blood and other extracellular fluids, but phosphate buffer overtakes the role of bicarbonate buffer at the level of renal tubules, and this is going to be one of our topics later on. So let's start with the first buffer, that is the bicarbonate buffer, and here is the equilibrium which I have written before. So we have an equilibrium reaction, equilibrium between carbonic acid and its gaseous form carbon dioxide, and we have the dissociation of carbonic acid to bicarbonate ion and hydrogen ion. So for both of these equilibriums we can write the equilibrium constant. So this is the equilibrium constant for hydration, K1, and if you remember our chemistry course, it is the ratio between the product of concentration of products and product of concentrations of reactants, and we can do the same for K2, which is the dissociation constant. So it's going to be hydrogen times bicarbonate over concentration of carbonic acid or carbon dioxide, whichever you write, you won't be wrong. So what can we do? We see now that we have a common component of those two equations, which is concentration of carbonic acid. So from this first equation we can write down the concentration of carbonic acid, and then we can exchange it in equation for the dissociation constant. So instead of concentration of carbonic acid, we can write down K1 times water times carbon dioxide. So we have two constants, K2 and K1, and what else we can do? We can consider the concentration of water as a constant value, because you know water is a solvent, and we learned that if the concentration of solvent predominates in a solution, its concentration is unchanged and it's constant. So if we consider that concentration of water is also constant, and we can combine K1, K2 and water into the third constant, for example K1 prim, we can get the following equation. So constant K1 equals hydrogen times bicarbonate over carbon dioxide. Now we can calculate the negative logarithm of this equation, and we're going to get the following equation. pH equals pK1 plus logarithm of the ratio of bicarbonate to carbon dioxide or carbonic acid. So if we consider that concentration of carbon dioxide is proportional to the partial pressure of carbon dioxide, and if we know that alpha is solubility coefficient, we can write it down in different ways. So whichever way you choose to express this equation, it's called Henderson-Hasselbalch equation, and it's actually the basis for assessment of hydrogen ions homeostasis. So under physiological conditions, so this is a practical application of this equation. We know that the average concentration of bicarbonate ions is 24 millimoles per liter in blood plasma. And we know that the physiological partial pressure of carbon dioxide is 40 millimeters of mercury scale, and alpha is 0.03, and pK for bicarbonate buffer is 6.1. So if we change all these numbers into our Henderson-Hasselbalch equation, we get 7.4. So it's exactly the physiological pH of extracellular fluid. Or we can do the opposite. So we can start from the final request. So if we need to have the pH of body fluids at the level of 7.4, and we know that pK for bicarbonate buffer is 6.1, then we can calculate the ratio between bicarbonate ion and carbonic acid or carbon dioxide, and it's 20:1. So we can conclude that for maintenance of normal pH, bicarbonate to carbon dioxide ratio has to be 20:1. And actually our body has to take all necessary corrections in order to keep that constant pH of body fluids. Bicarbonate buffer is considered to be the most important buffering system in the body, and there are several reasons for such a statement. It's very efficient. What does that mean? That means that the change in concentration of one component actually is almost immediately compensated by the change in the concentration of the second component. Let me explain this to you. So if we change the concentration of carbon dioxide, in order to maintain the ratio 20:1, we have to increase the concentration of bicarbonate ion, and vice versa. All other systems, and you will see that in a couple of minutes, all other buffering systems are actually cooperating with bicarbonate buffer. And moreover, carbon dioxide diffuses easily through membranes, so the effect of this buffering system is quickly reflected to intracellular compartments. Also, bicarbonate buffer is marked as alkali pool, or the first line defense for neutralization of acids in blood. The second very important buffering system is hemoglobin, and it acts together with bicarbonate ion in erythrocytes. And here are the components of this buffering system. So the acidic component of the buffer is deoxyhemoglobin, to which hydrogen ions are attached. And the conjugate base of this is oxygenated oxyhemoglobin. So buffering effects of proteins, thus hemoglobin as well, depend on the composition of those proteins, of the amino acid composition of those proteins, and the side chains which are found in amino acid sequence. So imidazole group in hemoglobin actually is capable of binding one more hydrogen ion. So the reaction, which is represented here, is shifted towards the right. So this reaction occurs in tissues with high concentration of hydrogen ions and low concentration of oxygen. So buffering capacity of hemoglobin is also increased because of the cooperation with bicarbonate buffer, and it will be explained a few minutes later. But one more information for you, and it's just to remind you about something that you know very well from the general biochem course. You know that hemoglobin is better buffer than other proteins, and the reason for that is a high content of amino acid histidine in the molecule of hemoglobin. And if you remember, the average content is 8 to 10% of amino acid content is actually histidine with its imidazole group, which is, as we explained, capable of binding one more hydrogen ion to hemoglobin molecule. When carbon dioxide is bound to hemoglobin, then we have the form of carbamino hemoglobin because it's attached to amino groups of proteins and hemoglobin. The third type of buffers are proteins. So definitely they are less efficient than hemoglobin. They are active in blood plasma. They are also active in erythrocytes, but with a less buffering capacity. The buffering capacity or action in interstitial fluid is negligible because of their absence, because there are no proteins in interstitial fluid. But proteins buffering systems are more important intracellularly, where they can neutralize or buffer hydrogen ions before they exit the cell. Which groups are responsible for buffering properties of proteins? Definitely those are side chains of acidic and basic amino acids like glutamic acid, aspartic acid, or those basic ones, lysine, arginine, histidine. And the last type is phosphate buffer, which consists of two components, dihydrogen phosphate and hydrogen phosphate as a conjugate base. And regarding the fact that pK for phosphate buffer is approximately 6.8, we can write the Henderson-Hasselbalch equation for phosphate buffer as well. So it exhibits a rather low buffering capacity in extracellular fluids and blood, for example, because the concentration of phosphates in extracellular fluids is rather low, not more than 2 millimoles per liter. So despite the very adequate pK of phosphate buffer, it has a low capacity due to low concentration of its components. However, phosphate buffer is much more important for buffering urine at the level of kidneys, because after the passage through tubules, water is reabsorbed, so the concentration of phosphate increases, and it increases almost up to 20-30 millimoles per liter. And it is very important because at the level of tubules, and you will see later on at the level of distal tubules, bicarbonate ions are fully reabsorbed or almost fully reabsorbed. So actually at this level, phosphate buffer actually overtakes the buffering function and the buffering role of bicarbonate buffer. So let's explain the role of respiratory system in regulation of acid-base equilibrium. So what is actually the role of respiratory system? Its role is to maintain a constant pH by retaining or eliminating carbon dioxide, of course, depending on the body's needs. This process occurs, of course, always in cooperation with bicarbonate buffer, and we will see with hemoglobin buffer as well. Let me remind you of something. Respiration, breathing, is controlled by certain respiratory centers in our hypothalamus, pons, and spinal cord. And they actually respond to pH change, and also they maintain the partial pressure of carbon dioxide at an average level of 5.3 kilopascals, or those 40 millimeters, or in a quantity of approximately 1.2 millimoles of carbon dioxide. So when there is a change, even slight change, respiratory system responds immediately to this change. But you have to take into account that the maximum effect is achieved only when both peripheral and central chemoreceptors are stimulated. So it will take from couple of minutes to couple of hours, 3 to 6 hours, to get back and to re-establish disturbed equilibrium. So anyway, before we move on to the exact mechanism of how lungs, bicarbonate buffer and hemoglobin buffer are cooperating in maintaining the pH, let me remind you of something that you probably know. Let's get back to our gases, oxygen and carbon dioxide, and let's see which mechanisms are involved in exchange of these gases in tissues and lungs. So let's start with oxygen. Oxygen is transported to tissues in two forms. Reversibly attached to hemoglobin in erythrocytes, and that is what we all know, in the form of oxyhemoglobin. Or certain smaller quantities can be transported as dissolved gas in erythrocytes fluid and blood plasma. So we can consider that conclude that hemoglobin is not only an oxygen carrier from lungs to tissues, but at the same time it exhibits certain very important buffering activities, and we will see that this buffering activity actually cooperates with the buffering activity of bicarbonate buffer in erythrocytes during the uptake or release of oxygen. So we will have uptake of oxygen, release of oxygen, and at the same time we're going to complete buffering activities. Regarding carbon dioxide, we know that it diffuses freely from lung capillaries into alveolar air. And alveoli have a very high capacity to eliminate carbon dioxide. Why is that so? Because the alveolar space is constantly emptied by experium, so there is always a concentration gradient between carbon dioxide in blood and less concentration of carbon dioxide in alveolar space. So carbon dioxide constantly diffuses from blood capillaries to alveolar space and eliminated from the body. Of course, that depends on the rate and depth of breathing. So oxygen is consumed during metabolism. This is what we all know for oxidative processes in mitochondria, thus forming carbon dioxide. We saw that in numerous metabolic pathways, and we have acidic organic metabolites of those metabolic pathways. So carbon dioxide, we will see in the mechanism, diffuses to blood again due to the differences in partial pressure due to this concentration gradient. And we mentioned that carbon dioxide can diffuse freely through membranes. So it's even stimulated by the existence of concentration gradient. So how carbon dioxide, once when formed in tissue cells, how can we transport carbon dioxide from tissues to lungs? So it can be transported in blood in several forms. So physiologically most important form is carbon dioxide dissolved in water in the form of bicarbonate ion in blood plasma and in blood erythrocytes. It can be transported in the form of carbaminohemoglobin, like we mentioned, in erythrocytes, but certain smaller quantities can be transported in the form of carbaminoproteins of plasma proteins. And of course, like oxygen, certain small quantity can be transported as dissolved gas in erythrocytes, fluid, and blood plasma. And again, like we emphasized for oxygen, these carbon dioxide transport forms are not only transport forms, but they rather represent dynamic system that is capable to buffer any change in pH of extracellular fluids, and also it participates in oxygen release and oxygen binding to hemoglobin. So let's explain the exact mechanism. So we have mentioned that the role of respiratory system is to maintain a constant pH by either retaining or eliminating carbon dioxide depending on the body's needs. This occurs together with the buffering activity of both bicarbonate and hemoglobin buffer. So, erythrocytes actually have a huge importance. They are one of the central points and central places where this maintenance occurs. And erythrocytes, if you know, they produce rather small, insignificant amounts of carbon dioxide, if any, because they lack mitochondria, so no aerobic metabolism is found here. So usually I like to explain the mechanism of this acid-base equilibrium maintenance by schemes, by figures, and I hope it's going to be simple and easy to understand. So I like to simplify rather complex things. So let's take a look at the following two figures and try to explain what's going on. So here we are. So we have tissue cells, we have blood plasma and erythrocytes. So during normal metabolism, we saw from the very beginning of this lecture that significant amount of acids is produced and significant amount of carbon dioxide is produced in oxidative processes in mitochondria. So you have the formation of a continuous concentration gradient between cells, blood plasma and erythrocytes. So the concentration of carbon dioxide is higher in tissue cells than in blood plasma. So carbon dioxide from metabolism will diffuse freely to blood. So when diffused to blood, upon diffused to blood, the concentration of carbon dioxide increases. And we mentioned from Henderson-Hasselbalch rule, if CO2 increases, bicarbonate has to be increased somehow as well. So we have to find a pattern, a mechanism, which is going to have the formation of bicarbonate ions as the final result to compensate the increase in carbon dioxide concentration. So let's follow this suggested mechanism. So due to the increase of CO2 concentration in blood plasma, CO2 can also diffuse freely through erythrocyte membrane and enter erythrocytes. Thus, we are increasing one of the reactants. One of the principles of Le Chatelier suggests that the equilibrium, carboanhydrase catalyzed equilibrium, will be shifted towards the production of products of the reaction. In this very case, carbon dioxide will be hydrated to carbonic acid and then dissociated to bicarbonate ion and hydrogen ions. Bicarbonate ion tend to exit erythrocytes to blood plasma, thus compensating the increase of carbon dioxide in blood. Again, if we get back here, if you have an equilibrium and if you are removing one of the products, which means bicarbonate ion, this reaction will be shifted towards formation of more products, thus shifting the removal of carbon dioxide from tissue cells towards the production of hydrogen ions. Now the increased concentration of hydrogen ions is overtaken by the next buffering system, and that is hemoglobin. So hemoglobin is gonna compensate this increase in hydrogen ion concentration in a way that hydrogen ions are gonna be bound to hemoglobin and oxygen released from hemoglobin. This is also stimulated by the fact that the increased concentration of oxygen will cause the diffuse process of oxygen to blood and from blood to tissue cells. So again, the equilibrium is shifted towards neutralization of hydrogen ions and production of deoxyhemoglobin. So if you notice that we did two things here at this first step. So we have removed carbon dioxide from tissue cells and we have supplied our tissue cells with fresh quantities of oxygen for oxidative processes. At the same time, we have compensated the increase in carbon dioxide in blood, plus we have captured those hydrogen ions in the form of deoxyhemoglobin. One more thing, in order to maintain electroneutrality, bicarbonate ions are exchanged for chloride ions. So chloride ions diffuses into erythrocytes, and this is marked as chloride shift or hamburger effect. Of course, if one can think that by this process concentration of chloride ions are going to constantly and continuously increase in erythrocytes, things wrong, because you will see from the next slide that the same quantity of chloride ions is going to be exported back to blood plasma. So the net concentration is going to be constant. So here we are again. So these hydrogen ions captured by hemoglobin are now transported to lungs and reaches alveolar capillaries. So we have alveoli, plasma, and erythrocytes. So I'm going to start with this. When we breathe in, we take a lot of oxygen into our alveoli. So we are making the concentration gradient. So oxygen diffuses from alveolar space to capillaries to blood and from blood to erythrocytes. Now we have the opposite situation. An increase in oxygen shifts the equilibrium of hemoglobin buffer towards its oxygenation, thus releasing hydrogen ions. Now hydrogen ions are overtaken by bicarbonate buffer in the reaction catalyzed by carboanhydrase, where hydrogen ions are going to be neutralized by bicarbonate ions imported now from blood into erythrocytes. The shift of the equilibrium is shifted towards the production of carbon dioxide, and carbon dioxide diffuses to blood due to gradient concentration gradient, and then from blood to alveolar space where it's eliminated by experium. And again, if you're removing one of the products of the reaction, Le Chatelier says equilibrium will be shifted towards more production. So when we are eliminating carbon dioxide, we're pushing the whole process towards eliminating more carbon dioxide. So again, we did the same. So now we got rid of the carbon dioxide, and that is the same carbon dioxide that we have uptaken from the tissue cells. But notice that this time hydrogen ions are captured inside the organism. So hydrogen ions are not excreted. We have only eliminated carbon dioxide and hydrogen ions are still here. It's going to be difference between the respiratory and renal system of regulation, and we will come to that in the second part of the lecture. So anyway, this was the first part. So we have elucidated the chemical buffers and their physiological role to buffer and neutralize excess amounts of hydrogen ions produced. And we saw in which way respiratory systems and lungs are involved together with bicarbonate buffer and hemoglobin buffer in maintaining the constant pH of body fluids. So in the next lecture, I'm going to continue with renal control of pH and acid-base equilibrium. And until then, stay healthy and stay safe, and we will see each other soon. Bye-bye.